Good morning.
Good morning. All right. So I think if I'm correct, we only have like 2 lectures to go, right? Including this one. So this Thursday, it is Thanksgiving, so we do not have class on Thursday. And next week, we will have the last lecture on Tuesday, I believe. And the next Thursday will be December 1st. So I think next Thursday, if I'm correct, let me double check. Oops. Yes, so next Thursday, the Thursday of next week will be the December 1st. So that's the time for us to present our term papers, right? Okay, so we're doing great, I think. Okay, so let's get started. Okay. Okay, so last time I very briefly started this topic, mapping segregation distortions loci. Okay, so this is the situation, right? So if this F2 population is following the Hardy-Weinberg theorem, and if we just check the genotype frequency. So for example, if this is a F2, so we should have 3 genotypes, right? A1, A1, A2, A1, A2, and A2, A2. Okay, so we have 3 genotypes at each genomic locus. So for example, here. So if this population is a real F2 and every single locus is following the Hardy-Weinberg, so we're going to see roughly this pattern, right? The 1 to 2 to 1 ratio. So this is 25%, this is 50%, and this is 25%. And of course, you're going to see some noise here. So you're going to see like it's not like perfect like horizontal line. It's kind of like noisy, right? But that's okay. So roughly, if every single locus is following this ratio 1 to 2 to 1 ratio, then we say we would conclude there's no segregation distortion in this population. However, so if you compare this plot with this plot, you're going to see difference, right? All right. The reason why is that because of the selection. So the selection, some kind of like selection force will produce the SD, which means the segregation distortion, and we want to find the loci that are subject to this the segregation distortion. We call this SDL. So we can develop some statistical genomic model to help us identify those loci with this pressure. Okay, so basically, okay, so these 2 graphs are a good example to show how we can form the hypothesis testing. So for example, this one is a perfect the non-hypothesis model, right? The non-model. So if everything is like this, then there's no selection stress on this on this population. So that's the perfect non-model. I hope to try to help you link this to something we are already familiar with. So in statistical test, we try to form a hypothesis, 2 hypotheses, right? To answer some question, we need to form 2 hypotheses, competing hypotheses. So for example, here, the question is, okay, is this F2 population subject to some like selection force to show the segregation distortion. So that's the null hypothesis. No, this population is okay. There's no selection force, and no segregation distortion is shown in this population. All right, that's the NUM model. Okay, however, if you look at here, so that's a perfect example to show the alternative model, so alternative hypothesis. So here, if we want to test any genomic loci that is any genomic locus that is a SDL, the segregation distortion locus, so we can compare this alternative model with versus the NUM model, right? So we just want to compare these two models. So obviously we see some deviation. For example, at this locus, we see some deviation from the classical 1:2:1 ratio, and we do not know if this deviation is a real deviation or it is just due to some random error, random noise. So we want to compare this deviation, which is shown in the alternative model, versus the NUM model. So we want to compare this with the 1:2:1 ratio. Sorry, and if the difference is big, so for example, if we get a large chi-square statistic, then this results will be in favor for the alternative model and against the NUM model, right? So this is the routine that we can use for testing this SDL. Any questions here? So the take-home message is actually we if we want to test the SDL, we want to identify the SDL. So we want so what we do is like we compare the alternative model here. This is the data we observe versus this NUM model. Okay, and I'm going to show you how we can set up a model to scan the whole genome to see if there are any SDL in this population. Okay. Right. So so for example, we have like 3 linked loci like A, B, and C. All right. So for this F2 progeny, we can check the genotype frequency for these 3 linked loci, right? And so suppose we use the pi A to pi A11 represent the observed genotype frequency for the first genotype, which is A1A1 at this locus for the first genotype, and this is the second genotype, and this is third genotype, and so on. This is for the B locus, C locus. All right, that's the data. And suppose in this F2 progeny, so we what we observed in this F2 progeny is like this. That's the data. All right, if this is the data, so what conclusion can we make? All right. All right. Do you think this is this ratio, this distribution is very close to the classical F2 ratio, like the 25%, 50%, and another 25%? Definitely, yeah. This is a question, right? So we don't believe this deviation is due to the noise. Maybe there is some selection force acting on this locus. That's why. So the B2, this genotype is in favor, but this genotype B1 and B1, which is significantly lower than 25%, right? So we believe this genotype probably is being selected against. All right, and okay, because so this data actually is showing like it's very likely that the selection is performing on this locus. But however, we also see some similar patterns, but not that severe pattern, right? On these two loci, A and C, this is because A, B, C are linked loci, so that's why. So even the selection force is only working on this locus. You also can see some like linked effect on these two flanking loci. Does that make sense? Okay. So this is why when you see, for example, this is the locus being selected either against or in a favor, then you're going to see some distortion as well on the adjacent loci. So that's because that's due to the linkage between this locus and the adjacent loci. Okay, let's see how we can use statistics to test whether any observed distortion are real or not. So suppose we are talking about like 3 linked loci, right? So on the gene on any chromosome, so N, G, and M. So suppose we only have the genotypic data for these 2 markers, M and N. But here in the middle, so we do not have any genotypic information, but that's okay. So before we take this class, maybe we will be mad if we do not have genotype like this position. But we are still asked to test this position, this locus, then we will become mad, right? But now after taking this class, you should feel very calm to handle this problem, because given this 2 markers, the genomic information about these 2 markers, and if we knew the genetic distance between these 2 markers and this unknown locus, we can easily infer the likely genotypes for this G locus, right? So this is what we can do by using the genetic statistical genetic model. All right. Okay. So since this is a F2 population, still for G locus we have 3 possible genotypes, right? Either the G1, or G1, G2, or G2. But to infer the likely genotype for this locus for any specific individual, so we have to use the information on these 2 markers. So this is how we handle this, right? I hope you still remember. So we have a transition matrix, which is like this and this. So if we consider the starting point is G, and we can think about like 2 Markov chain. One is on this direction, the other one is on this direction. Okay, then we can write this model. So this is the model to show the transition from G to N, right? Given, so suppose now starting from G, if G locus takes the I's genotype, the K's genotype, K can be 1, 2, and 3, right? So either one. So given the genotype at G locus, so what is the likelihood or the probability that M marker will take this genotype? So we're okay with this calculation. So we know this is the conditional probability, right? And this probability can be written in a transition matrix. Let me see if I have the transition matrix on the next slide. Yes. Okay. So this is the transition matrix I was referring to. All right. Okay. So suppose this is G11, G12, and G22. Okay. And the x-axis represent the M11, M12, and M22. Okay. All right. The way we use this transition matrix is like this. Suppose we want to calculate the probability that the marker M is 1, 1, given the G is 2, 2, right? So what's the probability? So how do we use this transition matrix to help us identify this transition probability? So here we actually need to use this row to represent the G22, right? And this column represent M11. So this is the probability for you to see this kind of transition. So the probability that you're going to see this genotype at G locus, but this genotype at M locus will be this, which means there are two crossovers, right, on both parental type. Okay, so this is how we use this transition matrix here. Any questions? Okay, perfect. Now we know this first transition matrix from G to M, and of course there's another way to model the Markov chain, right, on the right hand side. So given this G locus, what is the probability you see a different genotypes on the in the N locus? So this is the second transition matrix. So why we have two transition matrix, T1 and T2? Because the genetic distance between G and N and G and M are different, right? So that's why we use 1 and 2. And here we have like transition matrix 1 and the transition matrix 2 here, right? With this transition matrix specified, we can easily write the likelihood model. So this is the likelihood model, the core part of the likelihood model. So I'm going to show you on the next slides here. All right. All right. Okay. So suppose this is the probability. So part of the likelihood. So suppose in this individual J, and we see the genotype is 3, which is G22, right? And what is the probability that we're going to see like the first genotype for M, but the second genotype for N? So this is how we calculate this, right? Okay. And so any questions here to understand this part? All right. Okay, now this is the important thing, because here this is the proportion. This is do you still remember this? So this omega k, actually omega k represented the observed genotype frequency. So we have 3 genotype frequency, right? And this the summation of the 3 omega k should be 1. So this is the observed genotype frequency for the first one, the second one, and the third one. This is related to this graph. So here. So for example, this is omega 1. Let me use different color. So this from here to here is the omega 2. And this here to here, that's omega 3. So this omega 1 plus omega 2 and plus omega 3 equals to 1. That's the observed the real genotypic frequency at this locus in this F2 population. And actually, we want to test if this 3 is close to what? So can anyone tell me? So this actually is the alternative model. So we want to compare this with the alternative model. What is the alternative model here? Can anyone tell me?
Would it be 0?
No, we still we still need to see something like add up to 1, right? But if this is a non-model, which means there's no segregation distortion, so we're gonna see. So if this is the case, we're gonna have like omega-1 is roughly equal to 0.25, and this guy is roughly equal to this, right? And this guy roughly equal to this. So the non-model, which means like there's no selection force, and you do not see any segregation distortion. But this is the real data. If the real data is totally different from this ratio, then we're gonna reject the null hypothesis. We don't believe this this locus is a neutral one. We believe this locus actually is under some like selection force, and this locus is showing some deviation from this 1:2:1 ratio, and this one is a SDL. Does that make sense?
Yes, that's much clearer. Thank you.
Okay, sure. Yeah. So this is very fun. If you understand the underlying, it would be fun and easy to handle the analysis here. So that's why this guy is important. So we want to estimate. We don't know this yet, right? But we want to estimate the observed, the real, the genotype frequency for these two genotypes at locus G, and we want to compare. So from the data we can calculate, we can estimate these 3 numbers here. And once you have the estimation, you would like to compare these 3 numbers with these 3 numbers to see if they are identical, right? If they are identical, then you're going to accept the null hypothesis. Okay, this is a neutral loci. No SDL at this position. However, when you analyze your data, I'm going to show you how you analyze this data. So we have learned this before. We just need a little bit of modification. Then we can use that model to estimate the 3 brothers, and we're going to compare these 3 numbers to this 3 numbers to see if they're different. So that's why I see. Therefore, null hypothesis means like you calculated omega will be equal to phi. Phi is like this. Phi is a vector of this. This is your phi, which is the non-model. If your omega is equivalent to the phi, that means like this is not a SDL, no. But if this is not equal to this, which means like this is a great distortion, this is a great deviation from this vector, then you can claim you identified a SDL. Does that make sense?
Yeah.
Okay, cool. All right. Okay, so now we have this probability, right? The conditional probability. Then we can use this to construct a likelihood here. So this is the likelihood based on your data, based on your non-model. Okay, and we can rewrite this, the log likelihood. So once you have your log likelihood, so you should know what would be the next step, right? So we're gonna take the first and the second derivative to get the solution for this. So one of the target is to estimate this. So we need to estimate these numbers first. Okay, all right. And here we have some missing values here. So this is the missing. So the best way to do this is to use the EM algorithm, because this will help us to solve the problem very easily. So we have, this means like, so we do not know for each individual, let's say this is the J's individual, we do not know the genotype for this guy. So that's why this variable is missing. And but treat this guy as a missing value. If we treat this missing value as a non-missing value, then we can easily write the complete log likelihood. So we just assume this is a known value. Then we can write a simple complete log likelihood and use the maximum likelihood estimation to calculate the parameter, right? But in reality, this is not a value that can be observed. So how can we do that? So since this value is missing, we do not know the real value for this guy, but we can calculate the expected value for this guy, right? So this is the EM algorithm. So when we calculate the expected value for this delta, we can replace this missing value by its expected value. Then we can still use this complete log likelihood function and to do the MLE. That's the core part for EM algorithm. All right. Okay, so now we have this, and we have this complete likelihood, log likelihood. Then following the routine, so it's easy. We just calculate. So this is a EM algorithm. So we need to calculate the expected value at each step. And once we have the expected value, we replace the missing value by this expected value, and then we can get the maximum likelihood estimation. So this is how we calculate this guy. So we want to calculate this, right? This is the target. So then we can get the MLE for this guy. Yeah, don't worry. So this I will not test you on the finals on this in the final exam, but this is good for you to understand how the routine is implemented with this EM algorithm, and how we can estimate the parameters of interest and test if they are significantly different from the NUM model. Okay. All right, so this is the EM algorithm. So first, because we do not know this guy and we want to estimate this guy, right? So at the beginning, we can just randomly, arbitrarily give some like initial value for this guy. So if you do not know, so you can just give the even numbers to each of them. You can give like this 3 values to the omega 1, omega 2, and omega 3. So you don't know what they are, right? But you can just arbitrarily initialize them using some like numbers like this. And okay, then you can start your EM algorithm. So until this EM algorithm converged to some point, then you get the solution for these 3 parameters. Okay. All right. So once you get the estimation, the solution for this omega, so you can test whether this omega you calculated, right? This is omega 1, omega 2, and omega 3. So next you want to compare this versus what? Versus phi. Phi is the NUM model. In NUM model, you're gonna see 25.5 here, and another 0.25. You want to see if they are equivalent or not. So this is equivalent to what? Equivalent to like we just compare the likelihood based on the NUM model versus the likelihood based on the estimation from the data, right? So if these two are very close to each other, so this is the likelihood based on the NUM model, phi here, right? This is the likelihood based on the data. So if these two are very close to each other, so this value will be a small one, close to 0. What does that mean? Can anyone tell me?
It would be close to the null model.
Mm-hmm. Yes. So in this case, we're going to accept the NUM hypothesis, which means this locus is. So let me ask you. So if this is the case, do you conclude this locus is a SDL or not?
It is.
It is.
It is not actually.
Not
So
okay.
if it is a segregation distortion locus, then you're gonna see this one is not equal to the NAM model, right? If this is not a SDL, so means this, if this is a neutral locus, then you're gonna see this value is very close to the NAM model, right? Does that make sense?
Yes, yes.
Oh, okay.
Thank you.
Yeah, no problem. So here, actually, we compare. So the likelihood ratio test is always like this. So you have your NAM model, and based on your NAM model, the parameter, which is the 0.25, 0.5, and 0.25, right? You use that model to calculate one likelihood. That's the likelihood associated with the NAM model. And then you can calculate another likelihood, which is based on the parameter estimated from the data. And then you compare your the likelihood based on the alternative model versus the NAM model. If they are very close to each other, which means like your data is very close to the NAM model, then you're gonna accept the NAM model. Otherwise, if they are quite different, and if this is a large value, like let's say this is 100, if you calculate this likelihood ratio test, this means like there's a big difference between these two, which means your data is not following this NAM model, right? Okay, so this is how we calculate these two likelihood. Can you see the difference here? So can anyone tell me the difference between these two? So when we calculate this one and this one, let me use another color. What's the difference here? So actually this part is the same, this part is the same, this part is the same, this part is the same. Only this part is different. See, we use different value to calculate the likelihood for these two models, right? So what is this? This is NAM model, right? We use 25. These parameters here for the NAM model. But for the alternative model, this could be different. So maybe you estimate, when you use the EM algorithm, you can get something like 0.1 here, 0.8 here, and another 0.1 here. This is your estimation for this guy, right? So when you get this estimation, you use these parameters, estimated parameters, to calculate the likelihood for your alternative model. And this is always like this. You use this to calculate the NAM model. So if this is the case, I'm sure you're going to get a big difference between these two, right? So this value will be large, for example. Yeah.
What's T sub 1?
I'm sorry, say that again.
What is T sub 1?
You mean this?
No, I think he's referring to the left, next to the sigma.
Sigma.
Sorry, T sub 1?
T sub 1 is here, right? Are you
Yes.
Yeah, this is actually the transition matrix. So you still remember the transition matrix 1?
That's right.
Yeah, because for this G locus, we do not know its genotype, but we know the genotype at N and M, right? So we use the transition matrix. This is N, this is M. Then we can calculate the Markov chain transition probability. So this is T sub 1 and T sub 2 here.
Thank you.
Sure. Any other questions? Very good. Yeah, this is very difficult, complicated. So don't worry. So in the future, when you want to use it again, so probably you need to spend a lot of time to revisit this and make sure you understand every single details. But here, yeah, this is good enough. So I hope you understand the major steps here. So here we actually try to compare the likelihood from the non-model versus the likelihood from the alternative model. And this is how we calculate the likelihood for each of each of these two. And we're going to compare if they are different or not. So the difference between these two will be the test statistic, like the chi-square statistic. And if this guy is big, like 100, then this will this is the evidence, strong evidence against the null hypothesis. And if this value is very small, that's close to 0, which means the difference between these two are small, right? And this means like your data actually is supporting your null hypothesis, which means this is a neutral locus, not a SDL. Okay. Okay, let's move forward. Okay, so as I mentioned, this is the major part to test whether this locus is a SDL or not. So we have to first estimate this three, right? And there's a constraint for these three parameters. So these three values must add up to 1. Otherwise, there should be an error when you run the program, all right? Which means like there's a linear combination between the three numbers, so they are not completely independent. So that's why we can only calculate the variance covariance matrix with some constraint here. All right, this constraint will apply to the likelihood function here, right? And this is another way other than the EM algorithm. So we can still use some other method like the Newton's method or the Fisher's method to help us calculate the information matrix for this omega by doing the first derivative and the second derivative. So this is how we can calculate the variance covariance matrix for these parameters. Okay, so this is how we do this. Very complicated. Okay, all right. So I'm done with this part. Let's move on. Okay. All right. This is another way. There's another way that we can use for testing whether the G locus here is a SDL or not. This is another way. So suppose we have some other parameters. So those parameters are also missing. So, but we can treat them as some like imaginary variables that is associated with these three genotypes here. So suppose so we define relative fitness levels for the three genotypes here. So we used this is not so this is different from this. So we used omega k, right? But here I don't know. So we should use another name. But this, let's say the fitness for genotype 1, G1 is called the fitness 1-1. So this is different from here. So yeah, it's a little bit confusing here. So, but I try to make it clear to you. So we use this 1, 2, and 3 to represent what? To represent the genotype frequency for these three genotypes, right? But here we use the fitness 1-1. So this is different from here, right? And fitness 1-2 and fitness 2-2. The fitness for these three genotypes here. So if we put the fitness 1-1 as 1, then we can define the relative fitness level for the other two genotypes. So the second fitness level for the genotype 1-2, this one is 1 minus SH, S multiplied by H, and the fitness level for the G2 is defined as 1 minus S, and this S and H are defined as the selection coefficient and the degree of dominance. The S is the selection coefficient, and the H is the degree of dominance. Okay. All right. So we can define this 3 fitness levels, and okay, then we can for any F2 population, we can calculate the average fitness in this population. So because we know if this is a F2 population, right, before selection, they are supposed to follow this classic ratio, right? 1 to 2 to 1 ratio. This is 0.25, 0.5, and 0.25. Okay, now we calculate the average fitness for this population. And after the selection, we're going to see a segregation. Suppose selection is acting on this locus G, then after the selection, we're going to see the segregation, a different ratio, which is defined by this. This is the omega 1, right? The genotype frequency for the first genotype, second, and third. So we can calculate the genotype frequencies after the selection. And if we define S and H in that way, so there's a relationship between this S selection coefficient and the dominance coefficient by using this genotype frequencies here. All right. Okay. And then we can use the liability model. So we talk about this liability models to help us to analyze this data, right? The liability model here, how this model can work for this situation. So suppose if there's no selection, you're gonna see like the phenotype is normally distributed, right? Something like this. However, if this trait is being selected against for some like genotypes, for example, so you're gonna see like, so if anything like lower than this will be selected against it. So in the natural population, you do not see the individuals with phenotypic value in this region, right? What you can see in the population, the individuals survive from this selection will be this region. So if the selection force is very strong, so you're gonna see only this part of the individuals will survive. And if this is the selection model, so we can use the liability model to help us analyze this data. Okay. All right. So this is the phenotype value before the selection, and we can use. We are very familiar with this, right? So this is the additive and the dominance effect, potential additive effects and the dominance effect for this locus G. Okay, and this is the this is how we code for the additive effects, positive 1, 0, and -1, right? This is how we code the dominance effect. So we are familiar with this. All right, this model is very is straightforward. However, so we're gonna deal with this phenotypic value a little bit. So this is the phenotypic value before selection, but after the selection, only this part can be observed. So we want to calculate the probability that this phenotypic value is greater than some threshold here, and using this link function. Okay. That's the liability model. And all right, so then we can easily combine this liability model with the fitness. So this is how we define the fitness, right? The fitness is here, and we can combine this model to achieve this likelihood function. All right. Actually, we just try to formulate a problem which is originally a SDL mapping into a new problem which is about mapping QTL. So we just use what we are familiar and apply this familiar method to this new problem. So here we try to map the SDL, but we can still use the model for the QTL mapping to analyze this data. Yeah, this is a very smart way to do this. Okay. And so the same here. So we have the likelihood function. We do the first derivative and the second derivative, and we can get the information matrix. And also alternatively, we can use the EM algorithm to help us analyze the parameters and the variance. And then we can do the test. So here, we want to test whether this locus G is a SDL or not. So the null hypothesis is like the additive or the dominant effect is equal to 0, then this is a neutral loci. If this is the case, it is not a SDL. If alternatively, if either the additive effect is not equal to 0 or the dominant effect is not equal to 0, or both, then we can say this locus is not a neutral one. Put another way, this locus actually is indeed a SDL, which is subject to some selection force. All right. So based on this, we can calculate the two likelihood, right? So the likelihood based on the NAM model, this one, or the likelihood based on the alternative model, and we can compare this to whether they are the same or not. So this can help us calculate the p-value, chi-square test, statistic, and the p-value. Any questions here? Okay. Let's see. All right. And also, so this is even more complicated. So with the same data, we can test both. We can either map the QTL, potential QTL, and we can also test whether this locus is a segregation distortion locus. So we can map QTL and SDL simultaneously using a single model. Okay, so this is how we can do this. So now we have like a bunch of parameters here. So we have the omega, which is the genotype proportions, right? Frequencies. We want to test if this is different from this 25.25, 0.5, and 0.25. We want to test if this is equal to this. And we also want to test if gamma equals to 0, right? Gamma is like A and D, right? So we want to test whether this additive or dominant effect equals to 0 or not. So we can put them together. So that's the entire set of the parameters, including omega and eta here. And we still use this model, the normal density function here. And we can put all these parameters in a single model, and then we can estimate them and test them. So this is the log likelihood function. And we can follow the routine to estimate them. The first derivative, the second derivative, and we can estimate the variance for the parameters. And okay, so here we can test. So based on this model, let's see what we can test. First, we can test whether this is a QTL, right? So we can test whether gamma equals to 0 or not. Gamma is actually equivalent to the additive effects and the dominant effects. So this is the first test that we can do based on this model. Second, we can test whether this frequency equals to 0.25, 0.5, and 0.25, right? So this is mapping the SDL and we can also test them simultaneously. We can test whether gamma equals to 0 and whether this omega equals to that proportion, the 1 to 2 to 1 ratio. So in this case, we can do the SDL mapping and the QTL mapping simultaneously. Okay, so that's the Manhattan plot we are very familiar with, right? So that's the QTL mapping. So we can see where the genome in which genomic locus we have like some genes that are determine the variability of the traits, and if this trait is also being selected somehow, then when we do the SDL mapping, we're gonna get very similar results here, right? So you see the big segregation distortion along with this region. So if you compare this plot versus the first one, the QTL plot, you're gonna see usually this is the case. If in a F2 population, if you can identify some QTL, usually that locus is also being selected. The phenotype which is associated with that locus is also being selected. Any questions? Okay, we are pretty fast. So we're done with the chapter 11, and I'm gonna talk about the chapter 12. Any questions before we move forward? Any questions? Okay, yeah, let's continue. All right, so this chapter, we're gonna talk about how we do the QTL mapping in other populations. So far, we just focus on very basic population, right? Either the F2 or the BC, the backcross population. So in this chapter, we're gonna see if we can expand our technology to deal with some other breeding populations. Okay, so we have introduced this to you, right? I have introduced this population to you last time, the real population, which is the recombinant inbred lines, right? So we also okay. So this is the real population. How we can generate a real population here? All right, we start from two pure lines. For example, the rice. We have two rice pure lines, and if we cross these two parental lines, we get a F1 heterozygous. So usually F1. So we can treat this like completely like hybrid, and if we want, so there are two concepts. Two terms are very important in breeding, like this. So one is the heterozygosity level. And the other one is like the inbreeding coefficient. So for F1, we can say the heterozygosity level equals to 1, so completely heterozygous, right? And in this case, the inbreeding coefficient will be equal to 0. If the heterozygosity is equal to 1, then the inbreeding coefficient will be equal to 0. So that's for F1 generation here. Okay, now, so after the F1, so if we just do the selfing or just cross between the F1 progeny, we're gonna get F2, right? So in the F2, you're gonna see slightly different combinations due to the crossover recombination, right? And okay, so you cannot stop here. So if you want to develop a real population, you have to continue doing the cross between each generation. So if you do this, like you can get the F3 and F4s and go on. So after probably like 7 or 8 generation, you can get pure lines again. With the mixtured genes from these two parental lines, you're gonna get this. right? That population is called the real population. So something like this. So start from like two pure parental lines, F1, and then go through several generations. You're gonna get the homozygous lines, which is called the real population. And this process can be simply achieved by doing this. So in plants, usually this is very common in plants. So when you get F1, so if you can collect a lot of like gametes produced by F1, haploid gametes, and if you use some like a cytogenetic treatment for each of these gametes, just by doubling the genome, the haploid genome, you can generate the pure lines population. So we call this the doubled haploid population, or doubled haploid breeding. Right? Any questions here?
And would that be usually through like some type of mutagenesis?
Yes. So are you asking about like this process?
Yeah, whether it's dependent on just chemical mutagenesis or otherwise.
So yeah, that's a good question. So we need to do some literature research. So I think there's a lot of ways to achieve this, but yeah, I think that's the one of them. Yeah, yeah. So as long as we can achieve, like, for example, so this population is not achieved by the regular breeding program, right? So we need to use some kind of like chemical treatment or the cytogenetic treatment to make the haploid, the gametes, to double its genome, just like this, and then we have the diploid genome. Then this plant can be this line can be planned and can produce the individuals, right? So if we can generate a population like that, so this breeding population is called the doubled haploid breeding. I'm not expert in this field, but yeah, definitely. So if you can do a little bit like a literature search, you can find many ways to achieve this. Yeah.
Thank you.
Sure. Yeah, this is a very, very interesting field. So I think a lot of like cytogenetics and the breeders, they are using some advanced technology to achieve this population. It's good because it's fast. See, if you want to generate a real population, it takes time, right? It takes like a lot of time, like year, 2 years, at least like 7 years. So usually this is too long for a PhD, right? degree. But this can help you like just probably 1 generation. You can form your study population and do some analysis on this breeding cohort. All right. Any other questions here? All right. Okay. So the real population here usually generated from selfing, right? A generation by generation. And here, this is how we calculate this inbreeding coefficient. You remember? So this is so there's another term called the heterozygosity level. Heterozygosity level. All right, if the heterozygosity level equals to 1, then the inbreeding coefficient should be equal to 0. All right. Okay, let's look at the real population. So real one is like it will start from the F1 hybrid, as I mentioned. So F1 hybrid actually are totally like heterozygous. We can treat them as heterozygous. So in this case, the FT, we use FT to represent the inbreeding coefficient. So for the F1 here, this should be equal to 0, right? the inbreeding coefficient. Let's see if this equation makes sense to you. So if we are talking about F1, so the t equals to 1, then this thing will be 0. And this thing will be 0, then this whole thing will be 1, right? That 1 over 2 to the power of 0 equals to 1. Then 1 minus 1 equals to 0. Yes. So for F1, the inbreeding coefficient equals to 0. Yeah, that's correct. And then, all right, so let's say if you want to calculate the inbreeding coefficient for F2 population, then what is that? So when t equals to 2, so this equals to 1, right? And so the F2 should equal to 1 minus 1 over 2, which is 1 over 2. And as the generation by generation, you're going to see, where is it? Oops, sorry. The heterozygosity level is actually halved in each generation, right? And which means like your inbreeding coefficient will keep increasing, right? So after the 7th or 8th generation, let's see what happened here. So at the generation number 7, when t equals to 7, if you use this formula, you're gonna get like F7 equals to this. So usually in breeding, we use 0.98 as a cutoff. If the inbreeding coefficient is greater than this cutoff, then we can treat this as a pure line, complete homozygous individual. So this is for the real one. So this real one population is being created from constant selfing, right? And there's another way to achieve the real population. It's real 2. So this is generated from the brother sister mating, right? Usually, like for example, like in the animals, because in animals, breeding animals, you cannot do selfing, right? So so this is the commonly used way to generate real population in animals. So we use this brother sister mating. And so we always start from the second generation, right? And this is the formula we use to calculate the inbreeding coefficient in animal system. All right, and this is the function, just for your information. So this function actually shows in different like breeding programs. So how the inbreeding coefficients increase with the number of the generation. So if we do, so in plant, if we use the selfing, so it can achieve the real population very quickly, like maybe 7 or 8 generations. But in animal breeding, so if we can only use this mating system, then it takes a little bit longer to achieve the real population. Okay, and and I think in 1996, the McCain actually, they developed a model which can be generally applied to any breeding system, but with some specific mating system, breeding system, we have to use different parameters here. So for example, for brother sister mating system, this is the coefficient we need to use in this model. All right. Okay, now back to our business. So if we are dealing with like a real population, so how can we do the genetic mapping, the QTL mapping? We are familiar with this, right? So for the backcross design or the F2 design, so that's the model we're going to use. But here, since this is a real model, everything is pure line, homozygous. That means like we only have these 2 genotypes at each locus, right? We are losing the heterozygous loci because of the inbreeding coefficient that is very high. Any questions here? So this actually is good because we can increase the variance of this genotype variable. So once we can increase the variance of design matrix, then we can get a smaller reduced variance of the parameter estimation. Then we can get more accurate estimation. I'm going to show you. Yeah. And this is similar to the backcross design, but with larger variance here. Okay. So in real 1 and in real 2, instead of using the real genetic distance, we have to use a modified recombination fraction, or the genetic distance. Why is that? Because this is achieved not through a single generation, right? We achieve real population through maybe 7 or 8 generations crossing, right? Which means any crossover between two loci actually represent the accumulation of many generations of crossovers. So in the real population, after the 7th or 8th generations crossover, so for example, this is the A locus and this is the B locus. If there's only one generation, you probably see just one crossover between this A and B, right? But if there are multiple generations, you're going to see multiple crossover occurring between these two loci. So that will gives us the modified recombination fraction. So this is the original recombination fraction, the genetic distance between A and B. But in the real population, we have to use this modified. This modified will be larger than the original genetic distance between A and B. Any questions here? All right. So the transition matrix is the same. However, do you notice the difference here? So the transition matrix actually is very similar to the one we used to deal with the backcross design or the F2 design. However, we have to replace the RAB by this CAB, which is the modified recombination fraction. So this is the only difference here. And other than that, everything is the same. So we use the same model to do the QTL mapping. All right, so the same thing here. And all right, so this slides just show the difference between the real population and the regular mapping population. So as you can see, the CAB is equal to this, right? So if you simplify the equation on the previous slides, you're going to see this. And since the RAB is some value between 0 and 0.5, then when we look at the denominator, so this value must be less than 1, right? This value must be less than 1. And which means the entire thing must be greater than RAB. So this is, which means like the modified recombination frequency actually reflect the accumulation of multiple crossovers in multiple generations. Excuse me. And as I mentioned just now, because in this real real population design, we get better variance for this Z random variable equals to 1. And so this is how, because we are interested in the variance of the estimation of the parameters, right? Gamma actually is the additive effects and the dominant effects. So, and this variance of gamma reflect how accurate we can estimate this gamma. So we want this thing as smaller as possible, all right? And this variance actually equals to this. This is the residual variance. We cannot do anything with this one, but we can do something with this one, right? This is the variance of Z, this design matrix. So if we can increase this, here, then we can decrease this because this is in the denominator, right? So this is why this is the benefit of using this real population. Any questions here? So this could be a perfect question shown on your final exam. So I could ask you to compare different designs here. So you can So I would like to ask you to, for example, compare BC, F2, and real, and based on this, okay, and this, you can tell me which one is more efficient or more accurate in estimating the additive effects or the dominant effects. So this is the answer, right? So this slide actually gives you the detailed answer to that question. So because in backcross, you can calculate the variance for this C variable equals to this. In F2, because of this heterozygous genotype, you can calculate. So the variance of Z equals to this. This is greater than this. The F2 is greater than backcross, which means like the F2 design can give you more accurate estimation of the genetic effect. However, if you compare the real population with this two, so it's even better, right? Okay. All right. The double, the haploid, yeah. Yes. So okay. So probably this is the chemical that you can use, the colchicine. So this this chemical actually can block the formation of the spindle fiber, I believe. So this is the cytogenetic stuff. So for example, if either a haploid cell or diploid cell, when during the cell duplication, the first step for a cell to grow and duplicate is to double its genome, right? So double its genome, and followed by a division of the cell. Then you're going to see the same ploidy after the cell division. However, if you treat the cell with this colchicine, all right, the cell doesn't know that, right? The cell still like produce double the genome by itself, but this chemical can destroy, can block the formation of the spindle fiber, such that without the spindle fiber, the duplicated genome cannot be pulled apart to form two cells. So the doubled genome will stay in the same cell. So by doing this, you can double the genome and produce the doubled haploid populations. So this is one way. But I believe, so if you are interested in this, you can do some literature search and find some other technical ways to achieve the same thing. All right. Anyhow, let's go back to our business. We're not we do not worry about how people achieve this, but once they achieve this population, so we can do some mapping for this population, right? And this is the same with the real. But the only problem is like, because we achieve this by a single generation, so we do not need to use the CAB, which is the modified recombination frequency fraction, right? We just used our RAB original. So that's why this gives us the same resolution for the backcross design. Okay. I think I'm gonna stop here because of the time issue, and this is four-way cross. It's even more complicated. I don't want to discuss this part. I will leave this for you. So if you are interested in this, and if you have time, so yeah, you can go through these slides to see how we can handle four-way cross design and a full SIP family design. And so for the for the animal breeding, it's a little bit different, right? Because we cannot do selfing for animals. That's why you're gonna see some similar strategy. So usually, like in animal breeding, so we in plant we start from single parents, 2 bred lines, and then we do the F1 and F2 and go on, so we can achieve the real population. However, we cannot do this with animals. For animals, we start from 2 different breed. So breed A and breed B, we can treat this as like a parental 1 and a parental 2, just like plants. But in plants, we start from the individual plants. In animal, we have to start from a group of animal in breed A and in breed B, and then we can produce the F1 population, and then we do like the brother and sister crossing, then we can achieve like F2, F3, and so on. After maybe like 7 or 8th generation, we can get the real like population. All right, so I'm gonna stop here. Any questions for today?
No questions.
I just had a quick question about like the term paper. Is there like a page limit that you want us to reach on that?
Oh, you mean the PowerPoint, right? PowerPoint page number. So it's up to you. So if you can talk about the stuff very quickly, maybe you can prepare like maybe 30 slides. But if you want to like slow down, maybe 7 or 8 slides, because each of them only have like 10 minutes, right? 10 to 15 minutes. For a good presenter, we usually spend like maybe 1 minute on each page. So that means like the reasonable number of the PowerPoint page is about 10. So you spend about 1 minute on each page, and then you can finish this like maybe 10 to 15 minutes, and then followed by a 5 minutes discussion. Does that?
Sorry. No, I guess I was asking how long is the term paper?
Oh, the term paper. Okay. All right, so how about this? So this time I'm gonna reduce your burden, so you don't have to write your paper. You just need to prepare your PowerPoint slides in neat way. And so before December 1st, please send me your PowerPoint slides. That will be your term paper. I'm gonna review that. And the most important thing is how you present your PowerPoint presentation. Yeah, I have some like criteria. I will make up some like a list of the criteria for you. So that will be the criteria by which I can give you the scores for your presentation. Does that make sense?
Yeah, that makes sense. So so send it to you before December 1st. When would you be able to post the criteria?
So I'm gonna think about this during the Thanksgiving holidays and let you know as soon as possible. So don't worry, don't be nervous. So I just use very standard like way to evaluate your presentation. So I will not give you some strange like surprise criteria.
And then another question I had: when is the actual final?
The final is good question. So, all right, I'm gonna send you another announcement. Okay, so today is November 22nd. So Thursday we do not have class unless you want.
No.
Okay. All right. So we will have
exam is the December 3rd, the last time you said it, because I asked the same question.
Oh, yes. Okay. December 3rd, we will have our final exam. Yes, that's right. So we will have.
Wait, December 3rd is a Saturday.
Oh, sorry. 6th.
December 6th, perfect.
Sorry, yeah, December 6th. December 1st which is a Thursday, right? So we will have our presentation.
Okay.
Yeah. Okay, December 6th, we will have our final exam. And so I think we only have like 5 students here, so I don't think we will have. So on Thursday, December 1st, after our presentation, I'm gonna briefly talk about the final exam. I think by then I will be able to get the final exam test ready, so we can briefly talk about the scope. So this time we will be an open exam. So I'm going to give you, so everything can be find some. So for every question, you can find some clues from your lecture notes. So you can refer to lecture notes to find your answer. So that's the same part for everyone. But the different thing, so there's one part which will be different for each of you. So I'm going to generate 5 different data sets. And so you're gonna analyze your data set, specific data set, using some of my practice R code. And once you get the results, so you have to use your own judgment to make the conclusion based on the hypothesis testing. All right, does that make sense?
Yep.
Okay. All right. Any other questions for today?
Are we gonna have a dedicated review session for the final?
I'm sorry, say that again. So I cannot hear you. So
Sorry. Oh, yeah. Are you gonna have a review session for the final?
yeah. So after the presentation, how about this? So we can, if we have time, we can after the presentation, the term paper presentation, so we can have a discussion, the review session, or if on that day we do not have a lot of time, so maybe we can have another review section on Monday before the Tuesday's final exam. Does that make sense to everybody? Monday, December 5th. That's the final week, so I don't think like you have any other class schedule. So we can talk about this on Thursday, December 1st, if another review session is necessary. Yeah, if everybody are happy with that, so we can have another discussion.
I guess just a quick question. I mean, would this upcoming Tuesday also be an option? The 29th.
29th. Yeah, so that's a lecture day, right? 29th.
Yeah.
Yeah, we will have our last lecture on 29th. Yes. So the plan is like, so as you can see, I have already uploaded the lecture slides to the Canvas. So for the chapter 13 and on, so that's the totally application. So we are done with the theoretical parts already. For the later chapters, we just keep applying those method theorem to analyze different problems. So I'm going to use the last lecture to finish everything very fast.
If we have questions, like we have the time on Tuesday, can we ask you questions?
Oh, definitely. Yes.
Perfect. Thank you. Thank you so much.
Sure. Yeah. Any other questions? Oh, by the way, so I think I got an email from campus department evaluation department. They said like the class evaluation is open. Yeah, if you have time, please, do that and let me know if anything I can improve and do better in the future. Thank you.
All right. Thank you.
Thank you, everyone. Okay, if you do not have any questions, we're going to stop here. Right. Bye. See you next time.